Hi everyone, and welcome to a video on AQA A-Level Biology Carbohydrates. So this video, the aim of this video is to give you a detailed insight into what you need to know about carbohydrates for this course and why carbohydrates are actually useful. So when we talk about carbohydrates, we refer to sugars really, because complex carbohydrates can be broken down into sugars, simple sugars, and simple carbohydrates usually are sugars. So when you think of things like glucose, etc., they're sugars. Things like starch, glycogen, they're more complex carbohydrates, but they can still be broken down into those simple sugars. That's why the term sugars is a kind of a general term used for describing more so monosaccharides and disaccharides, but they can also be used to describe more complex carbohydrates like polysaccharides as well. So that brings me on to my next point. Carbohydrates are molecules that are made up of carbon, hydrogen, and oxygen, and the monomers of carbohydrates are called monosaccharides. The dimers of carbohydrates are called disaccharides, and the polymers of carbohydrates are known as polysaccharides. Now, examples of monosaccharides are things like glucose, fructose, galactose. Examples of disaccharides include maltose, sucrose, and lactose. And polysaccharides involve things like starch, glycogen, cellulose, so on and so forth. They're examples of polysaccharides. Now, for your syllabus, what you need to know is that there are two different types of glucose, what we call isomers. And isomers are just compounds that have the same molecular formula but a different kind of structure in space, so a different structural formula. And there are two isomers that you need to know about for glucose, one of them being alpha glucose and another one being beta glucose. And glucose is what we call a hexose sugar. We call it a hexose sugar because it comprises 6 carbons, and the general formula for glucose is C6H12O6. So you can see that it involves 6 carbons. So in its cyclic form, that's just as a molecule that has its ends reacted together to make a cycle type molecule. So the cyclic form of glucose looks like this. Now, there's a reason why I haven't drawn the two atoms, groups, that come off this carbon at the end here. And the reason is because there are two forms of glucose. So the one I'm drawing right now, I'm going to label alpha glucose. And the reason it's alpha is because the OH group is at the bottom. And an easy way to kind of remember this is that alpha kind of looks like what you draw as a fish, right? So fish also live in the sea, therefore the OH is at the bottom. That's a way that I like to remember it. And I think it's a really useful way of remembering which way the OH goes around. Because the alternative, so the isomer of alpha glucose is this molecule that we call beta glucose. Again, you'll notice that the OH group, HOH group on the left side, they're the same on both molecules. So this bit here and this bit here are exactly the same. It's just the OH group on the right hand side of this molecule that's changing. And we call this beta glucose because the OH group is at the top. Again, a nice way of remembering this is that beta, kind of if you have two lots like this, it looks like a butterfly. Butterflies are in the sky, so the OH group is at the top. The other way of remembering it is that B for bed, your bed's upstairs usually, so the OH group's at the top. Either way, they're both kind of like neat ways of remembering it. So those are the two isomers of glucose that you need to know about. Now, glucose can also be written slightly differently. So you might come across this sort of notation of glucose. Here I'm just going to draw alpha glucose. But the reason, so that you might come across this, and this is completely normal. It's just the way chemists, etc., and bioscientists usually write things. It's just shorthand for this. It doesn't involve the carbons or the hydrogens, the groups that we don't really care that much about, because these are the two terminal groups that will be reacting with things. They'll be involved in hydrolysis and condensation reactions, depending on where the molecule is. So let's have a look at disaccharide formation. So like I mentioned earlier, some examples of disaccharides are maltose, sucrose, and lactose. And disaccharides are formed by the condensation reaction of two monosaccharides. So what is a condensation reaction? Well, a condensation reaction is where two molecules react and a water molecule is removed from the two molecules, basically. So the reaction causes a water molecule to be lost, and that's what we call a condensation reaction. It forms a bond, and in this case, in the case of sugars, the bond that you get is what we call a glycosidic bond. So if we have a look at two monomers reacting, let's say we react two alpha glucose molecules. If we react two alpha glucose molecules, what we'll end up getting is maltose. So let's have a look at that reaction. So we've got one alpha glucose here, and let's get another alpha glucose here. I've drawn them slightly weirdly, but you'll still be able to see what's going on. So that's not a problem. So what happens is the molecule of water is taken out from here. These two react and this forms a molecule of water that's taken out. We end up with maltose, which is the same thing, but just I'm going to draw it down here just because I have a bit more space. But these two molecules are, like I said, alpha glucose. And what you form is maltose, which looks like this molecule here. And you can see that what's happened is this kind of oxygen bridge has formed which connects the two molecules together, and this is what we call our glycosidic bond. So the products of this reaction between our two alpha glucose molecules is maltose and also a molecule of water. And that's essentially what a condensation reaction is. Now, the opposite of a condensation reaction, so if we wanted to split this disaccharide up, the way we do this is by putting a molecule of water in, and putting a molecule of water in kind of reverses this entire thing. So it goes from maltose and our glycosidic bond. If we chuck some water like that, it splits this glycosidic bond, breaks the glycosidic bond, and splits the molecules apart to give their monomers, which are the two alpha glucose molecules. And this happens whenever we want to produce disaccharides or hydrolyze disaccharides into monosaccharides. So the types of disaccharides that you need to know about are sucrose, lactose, and maltose. And you need to know how we get those. So to get maltose, like I showed you, you need alpha glucose. You need two alpha glucose molecules to give you maltose. For sucrose, we're going to use glucose and galactose. That's going to produce lactose. And for sucrose, we need glucose and fructose to give us sucrose. And that's all there is to monosaccharides and disaccharides. And in the next tutorial, I'll show you what polysaccharides are, how we make them, and their uses. Thanks for watching, and I'll see you in the next video.